Hi, I'm James Campbell. Thanks for joining us today. I'm delighted to be with you to introduce you to Patras, introduce you to our technologies, and introduce you to why we think we've got the potential to develop fantastic new therapies to treat a range of really difficult to treat cancers. I'd like to take this opportunity to acknowledge the traditional owners of the land from which I talk to you from, the Wathaurong people. Let's move to the presentation. Safe Harbour statement, which I'll take as being read. In broad terms, the summary of Patras. Patras is developing a unique antibody platform. We call it our deoxymab platform, which are a group of cancer-targeting antibodies which can cross the blood-brain barrier and block DNA repair, leading to the death of cancer cells, regardless of whether they're inside or outside the brain. Deoxymabs have a range of different applications. Originally, we were looking at them as single agents, but the more work we've done with deoxymabs, it's very clear that there's great potential combining them with other therapeutics and using them as targeting agents for things like antibody drug conjugates or diagnostics. As I said, deoxymabs can cross the blood-brain barrier, which means there's potential there for these to be the first therapeutic antibodies for cancer that can cross the blood-brain barrier. Deoxymabs deal with attractive markets. PARP inhibitors are a class of DDR inhibitors, just like deoxymabs. Market sales there north of US 2 billion last year. And the deal space, certainly around what's called the DDR, or the DNA damage repair space, or the antibody drug conjugates, is running very hot. Multiple multi-billion-dollar deals in the last year. Patras has built an outstanding team. A team of credible people with drugs developed, drugs approved, assets transacted, and companies sold. I think if you look at the board and the management team, you'd be impressed. And finally, we've got a strong balance sheet at the moment. As at the 30th of September, 9.8 million of cash, 1.2 burn in the previous quarter, and we're funded for our lead agent into the clinic at the end of next year. Deoxymabs are the first therapeutic antibodies which target the DNA damage repair system, or the DDR. They are antibodies which bind to the DNA which is released from dying cancer cells. They've got a unique combination of properties. Deoxymabs are cancer-seeking. They are pan-cancer. They will localize very, very quickly towards cancers both inside and outside the brain. Once they're in the tumor environment, they are cell-penetrating. The antibodies move inside the cells, then they move inside the nucleus, the nuclei of those cells, where they block the DNA damage repair machinery, causing cancer cell death. As I've alluded to, deoxymabs can cross the blood-brain barrier, which opens up some really exciting opportunities for brain cancers, which, as you all know, historically have been very, very difficult to treat and have poor prognoses for patients. Preclinically, we've seen the deoxymabs are safe and there's very, very little effect on normal healthy cells. And intriguingly, there is some clinical experience, a phase 1 study with an early progenitor of our deoxymab technology, and certainly there were no tox issues when that was looked at about 25 years ago. The unique attributes of deoxymabs mean that we can use them in a range of different ways. As a single agent, they are effective at killing cancer cells that have these DDR mutations. BRCA is a classic example of a DDR mutation I'm sure you're familiar with. And what we've consistently seen with our lead agent, PAT-DX1, is that we can get a 50% increase in median survival in very difficult to treat cancers like triple-negative breast cancer, glioblastoma, and pancreatic cancer. We see this as a very positive sign. We also know that in the absence of a mutation status, by combining deoxymabs with other DNA damaging therapy like radiation or chemo, then we also get enhanced effects. For instance, a study we did at Yale showed that combining DX1 with radiation improved survival 3-fold compared to radiation alone, which is a very positive outcome. Finally, we can use the fact that deoxymabs localize at tumors and localize inside cancer cells to harness them for the delivery of payloads. For instance, we have been looking at antibody drug conjugates, and we recently reported a 99.7% tumor growth inhibition in a breast cancer model. There's imaging opportunities, and it's public knowledge that we're collaborating with Imagine on novel imaging agents. And there are a range of folk globally who are very interested in the intracellular delivery of payloads. So there's some conversations happening there as well. What are these? What sort of results do we see? I'm not going to dwell on these data, but very consistently in difficult to treat cancers like glioblastoma, triple negative breast cancer, pancreatic cancer, we see 45, 47, 50% increase in median survival. Strong results. Why is it a good time to look at Patras now, if we're still a year away from the clinic? Very interestingly, there's a dichotomy in deals between biologics and small molecules. We've looked at the data, and it's very clear that 60% of biologics transact before they get into the clinic. It's the obverse for small molecules, where you see 60% of them usually transacting after phase 2. We know that the demand for biologics is higher. Over a 2-year period, we saw more than twice as many deals in biologics compared to small molecules, and the deal value is significantly higher as well. What do those deals look like? Large numbers. We don't have time to look at them all, but these are preclinical deals, and their numbers, around north of $1 billion. That gives us a bit of enthusiasm for the state of the market at the moment. We've talked about the intellectual property and the intellectual capital of the company. Just as important as the human capital. Here's our board of directors. I'd encourage you to look at the CVs and the expertise there, particularly of our US-based directors who have been involved in the development, the approval and the licensing of a number of drugs. The snapshot, as I said to you earlier on, we've got a strong cash position at the moment. We're burning 1.2 per quarter. The share price has seen an increase over the last 12 months, and we feel that the next 12 months will be even more positive. So looking ahead, what are we doing at the moment? Great question. We're currently completing our engineering run for PAT-DX1. That's the asset we'll take to the clinic at the end of next year. That will be moving into toxicology in Q1 and Q2 of next year. We'll prepare the Australian IND application and hope to be launching the phase 1 in Q4 of next year. Simultaneously, we're working on platform extension. We're doing a lot of work around DX3. We are very excited by the data we're seeing with antibody drug conjugates, and there are a range of different collaborations that we'll report on when the time is right. So I hope I've been able to convey to you in this brief 8 minutes the enthusiasm that we have for Patras. Hopefully you're keen to find out more, so please feel free, reach out. I'm always happy to talk in perhaps a bit more detail than we had time for today. Thanks very much. See you.